about the P50, as I mentioned, P50 is a pressure of oxygen at which you have a half saturation, 2% saturation of your molecule. And as you can see here, as I mentioned, the form of the curve for myoglobin is hyperbolic. I mean the black curve. The blue curve is for hemoglobin. I will talk about the curve about the hemoglobin. But you should know that, as I mentioned, myoglobin is not a suitable molecule for transpiration of the oxygen. It's considered as a reservoir, okay, for the some days that you are at an emergency situation, okay? Whenever you are at risk of actual death, I mean that, for example, whenever you encounter with a very, very low pressure of oxygen, now the myoglobin can release the oxygen, okay? Because it has a very high affinity for the oxygen. So it's better to replace the myoglobin to the hemoglobin for transpiration of the oxygen. About the hemoglobin, this is an overview of the hemoglobin. This most simplest question is the number of the chain that's present in a structure of the hemoglobin. How many polypeptide chains do we have?
2.
4. Okay? Alpha 1, alpha 2, beta 1, beta 2. Another question. How many heme groups are present?
2.
Why 2? You get stuck in a 2. No. Depend on a number of your chain, you have a heme group. So for each chain, you have a 1 heme. So how many chains present? 4. How many heme group? 4. You know, each part of hemoglobin is like, for example, this is myoglobin. It's similar to myoglobin, similar to myoglobin, similar to myoglobin. Okay? Recall that. Myoglobin فقط 1 chain. فقط 1 heme. Okay? So, about the hemoglobin, because you have a 4 chain, so how many heme are present? 4. Another question. How many oxygen will bind to the myoglobin? To each molecule of oxygen, to each molecule of myoglobin. How many oxygen?
4.
It's better to actually ask in another way. How many oxygen can bind to the 1 heme? 1 oxygen. Okay? For each, please pay attention, for each myoglobin, for each heme group, فقط 1 oxygen here. فقط only 1 oxygen. So, how many is actually chains present in myoglobin? No, wait. I want to repeat actually some important point. How many chain is present in myoglobin? If you open the structure of myoglobin, single, excellent, only 1. Okay? How many heme group present in myoglobin? How many heme group do we have? فقط 1. I'm talking about myoglobin. So I want to repeat my question. How many chain is present in myoglobin? 1. How many heme is present? 1. How many oxygen can bind? 1. What about hemoglobin? How many chains are present? 4. How many heme group are present? 4. So we can say that how many oxygen molecule? 4. Is it clear? So each hemoglobin molecule can transfer 4 atoms of the oxygen. As I mentioned, this is a tetrameric. What does it mean, tetrameric? Tetra yani? You have a mono. Mono yani? Di. Tri. Tetra. Penta. Okay? So we say that hemoglobin is a tetrameric molecule. Mer means that wahda. Units. Okay? Tetramer yani 4 units is present. Okay? 2 of them is alpha and 2 of them is beta. And at the middle of each chain you have a heme group. And as I mentioned, each heme group can bind to the oxygen. Where we can find the hemoglobin? In which cells of the blood?
Red cells.
Excellent. So inside the RBC you can find the hemoglobin. What is the role of the hemoglobin? Transport of oxygen. After taking the deep breath, you take all of the oxygen from the lung to the tissues. And each, as I mentioned, hemoglobin can carry 4 more molecules of the oxygen. I want to explain the role of the hemoglobin. Okay, for example, here is lung. Okay, I take a deep breath and I take all of the oxygen. You know, you have some active tissue, like for example, muscle, like a liver. Okay, at first, you should know that. I think a little you're familiar with the metabolism. What is metabolism? Is it catabolism and anabolism, for example? For example, you want to digest the glucose. Okay, this is known as a metabolism, and the pathway, I think you're familiar with the glycolysis. You are familiar with glycolysis? Glycolysis finally convert to the pyruvate. Okay, are you familiar with this pathway?
And then it goes to Krebs cycle.
Excellent. We have a Krebs cycle. Excellent.
38.
Yes.
Glucose break ATP into glycolysis and then
Excellent.
pyruvic acid into the Krebs cycle and other cycles for breathing and digestion.
Excellent. These are examples of metabolic pathway. Okay. And also we have a metabolic pathway for protein, metabolic pathway for amino acid, okay, for lipids and other molecules. I didn't want to focus on details. I want to just generally show that as a result of metabolism, you produce a carbon dioxide. Am I right? CO2, carbon dioxide. Okay? For example, you have an active tissue, like an active muscle. You use, for example, glucose, and finally, you produce the CO2. But another point, this is actually new, actually issue. Not only after metabolism CO2 produced, but also your environment became acidic. Acidic yani proton produced. Okay? So I want to repeat. After activity of the body, after activity of the tissue, after metabolism, CO2 produced here. And also proton produced here. So I want to ask a question. This is full of proton, H proton. If I didn't actually collect this proton, if I didn't collect, what happened? What is the disorder? I'm sure Dr. Imam will explain. What is the name of disorder? After accumulation of the acid, accumulation of the proton, H. Acidosis. Do you remember? If you didn't control this H is full of acid. Our body became fully acidic. Okay? Hold from this point. CO2 and H. I want to turn back to the role of the hemoglobin. Okay. I'm hemoglobin. I'm in the lung. Okay. After taking deep breath, you bind to all of the oxygen. How many oxygen bind?
4.
4. Excellent. So I want to explain the actually different type of the structure. I want to explain the next slide. You should know that 2 forms of the hemoglobin is present. Okay, before everything. For example, consider me as a hemoglobin. 1, 2, 3, 4. Okay. I have a 2 form. One of them is R form, open structure with a high affinity. For example, if this is oxygen, I have a very high affinity. I'm Bahil now in this form. I have an open structure, and now I'm R form. Okay. Where we have an R form? In a lung. The pressure of oxygen is very high. Am I right? In a lung, the pressure of oxygen is high. So I can take all of the 4 oxygen. So now I'm full of oxygen. Okay? What is my role? I want to transfer to the tissue. For example, I want to reach to the muscle. What is the role of hemoglobin here? I want to release the oxygen. Am I right? Because you have active tissue. For example, you have a severe exercise. You are running. You have to provide the oxygen. Am I right? So, you release the oxygen. This is the first role of oxygen. Releasing at first. Releasing oxygen. And another role. Not only it releases oxygen, it takes a proton and take a carbon dioxide. Am I right? And it's again turn back to the lung, release the carbon dioxide. Do you understand the role of the hemoglobin? So we have a 2 form. I want to explain the 2 form of the hemoglobin. In a lung, as I mentioned, you have a very open structure. This is the R form. So if I ask you, in a lung, what is the form of the hemoglobin? R. Okay? Open. But whenever it reaches to the tissues, after releasing of oxygen, one by one, the structure will close. And the shape convert to the T form. And T form has a low affinity for oxygen. Didn't like oxygen, it's clear. Because I want to release my oxygen. If I have a high affinity, can I release oxygen? No, like in myoglobin. Myoglobin cannot take, okay? But hemoglobin, when it reaches to the tissue, release the oxygen and close the structure. About the bond between, for example, alpha, beta, okay? About the bond between alpha, beta unit, this is a salt bridge, okay? I want to ask you, recall the name of the ionic bond. Salt bridge, electrostatic interaction, they are synonym. Salt bridge يعني interaction between positive, negative. يعني ionic interaction. All of them have same meaning, okay? So whenever you have actually closed structure with a low affinity to oxygen in a tissue, you have a T form of the hemoglobin. So this is a structure of the hemoglobin we talk about. It's present as a 2-dimer, alpha, beta. What is important in this slide? You should know that, as I mentioned, I focus on an ionic bond between dimers, you know? 1 dimer from 2 dimer. For example, this is 1 dimer, another dimer, okay? This is a salt bridge, ionic bond, okay? But inside the dimer between, for example, alpha and beta, you have some hydrophobic interaction. But what is important for me? Between dimers and especially ionic bonds. Yani between positive, negative charge. We talk about this point. And these are 2 forms, as I mentioned. we want to review. T form, T is not originate from the tissue, but I want to help you to memorize it better. T yani tissue, if you want to memorize it better, okay? T means thought, but near tissue we have a T form. But what is the characteristic of T form? This is a oxyform or the oxyform. Now you can memorize it better. The oxyform. So another question. T form has a low affinity or high affinity? Excellent. Low affinity is in the near tissue. You didn't like oxygen because you want to release them. Okay? So you have actually T form. And in contrast, you have R form. High affinity in a lung, and also this is the oxy form of the hemoglobin. I want to turn back on oxygen dissociation curve. We talk about this curve, but I want to repeat this point.
Doctor,
As I mentioned, yes.
what is mean?
R means relax. No. Sometimes mention relax, but R rigid, not rigid. Okay. Rigid yani bakhil. You know, I want to help you to better memorize it. R actually originated from the T and R from the relax and thought and tense form. Actually, I put it here. T means that thought, and R yani relax. But relax, maybe you think that relax has a low affinity, but R is a very bakhil molecule. Okay, high affinity. But about the origin of the word, the main origin, relax and thought. Or tense, in some references mentioned that tense. We talk about the P15. As I mentioned, this is a pressure of oxygen at which we require, the pressure of the oxygen we require to reach to the half saturation. Half saturation yani 50% of the saturation. About the point is that this is the sigmoidal curve. The most simplest question, as I mentioned, if you draw the percent of saturation against the pressure of oxygen for hemoglobin, this is hyperbolic. But about myoglobin, sigmoidal. What about the myoglobin? Hyperbolic. So, I want to repeat my question. The saturation curve or oxygen dissociation curve of the myoglobin is hyperbolic, for hemoglobin sigmoidal. But what does it mean, sigmoidal? I want to explain the him-to-him interaction. You know, whenever you are in a lung, okay, and as I mentioned, how many oxygen will bind to the hemoglobin? 4 molecules. You know, we have one definition known as the him-to-him interaction. Whenever 4 molecules of oxygen bind to the hemoglobin, the affinity of the first and the last one is not equal. You know, they bind to the hemoglobin with a different affinity. Okay, and this process known as the him-to-him interaction, yani binding of one oxygen can affected by the presence of first, second, or third of oxygen. Okay, yani whenever you have a interaction between units of molecule, especially for the hemoglobin, you say that him-to-him interaction. In some slide I explain him-to-him interaction. I didn't put the him-to-him interaction. Okay, I actually removed the explanation here. Him-him interaction, as I mentioned, the oxygen dissociation curve of saturation, pressure of oxygen for the hemoglobin is sigmoidal because it resembles the S letter is known as a sigmoidal. And we say that this is a heme-to-heme interaction. The net effect, that's the affinity of the hemoglobin for the last oxygen bind is 300 times greater than the first oxygen. What does it mean? For example, after releasing all of your oxygen, okay, I want to explain the last line. The net effect is affinity of the hemoglobin for the last oxygen bond is 300 greater than. What does it mean? Okay, I